Single-cell RNA sequencing analysis reveals which genes are active in individual cells, helping scientists understand cellular functions. However, it doesn't explain why certain genes are active. Gene activity is controlled by regulatory elements in DNA, which can be studied using single-cell ATAC sequencing. Single-cell ATAC sequencing, it identifies open chromatin regions, indicating where gene regulation occurs. It doesn't directly show which genes are being used. To bridge this gap, scientists integrate both single-cell RNA sequencing and single-cell ATAC-seq. Using Seurat, this allows them to integrate gene activity, link chromatin accessibility to gene expression, and label single-cell ATAC cells based on single-cell RNA-seq data. And that's exactly what we are going to do today. Without further ado, let's get started. Firstly, we upload our libraries. As you can see, we have scNAC, Seurat, genomic ranges, ggplot2, patchwork, Seurat data, ensemble database hsapiens, cowplot, and biovsbase. Let's run our code. Next, we load our dataset, which is PBMC, or peripheral blood mononuclear cells, a type of blood cell essential for immune responses. So, we write ppmc.rna and then load data, and we put ppmc multi-ome ppmc.rna, and here the ATAC data. The same, we write load data and we put our data but .atac. So as you can see, we have two datasets represent different types of data: gene expression data from rnaseq and the chromatin accessibility data from ATAC sequencing. Now let's convert the RNA assay to assay 5. As in Seurat, an assay represents a specific type of molecular data stored within the object. For example, RNA expression, chromatin accessibility, protein levels, and others. The Seurat package supports multiple assay types, and the assay 5 is likely a customized or optimized format for this analysis. This conversion might be necessary for various reasons, such as enabling specific functionalities, improving data processing efficiency, or applying advanced methods for differential expression analysis. Now let's subset the data, which means removing filtered annotations, as it's common to perform initial quality control where cells that do not pass quality check. For example, cells with too few reads or too many reads or other issues. These cells are excluded from the analysis. So, as you can see, ppmc.rna, we put RNA here, and then as ppmc.rna, we put RNA, and class, we put assay 5. And then we repeat the quality control steps here for RNA and ATAC by making subset. And then Seurat annotations, filtered for both data. Now let's normalize and pre-process the data. So we write the ppmc.rna, we make normalized data, ppmc.rna, and then we can find the variable features. Here we normalize the rnaseq data so that each cell's gene expression is comparable. This help accounts for technical variation between cells, ensuring that differences in gene expression are due to biological signals, not technical noise. We can also identify genes whose expression varies most across cells. So, for example, as you can see, find variable features ppmc.rna, and also we can scale the gene expression data so that each gene has a mean of 0 and a variance of 1. This helps with normalization, ensuring that genes with higher overall expression, which could dominate the analysis, do not overshadow those with lower expression levels. In this case, we write scale data and we put ppmc.rna. In addition, we can also run principal component analysis, which is a technique that reduces dimensionality of the data, capturing the most important patterns of variation in fewer dimensions. In this case, we run PCA function. Not only that, but we can also run uniform manifold approximation and projection, which is UMAP. In this case, we write ppmc.rna and for dimensions from 1 to 30. So this indicates that UMAP is using the first 30 principal components. Now let's add gene annotations to the ATAC-seq data. We make a variable called annotations, and then we use the function getGRanges from ensemble database, and we put ensemble database H sapiens or Homo sapiens version 86. As an ATAC-seq data, it's crucial to associate peaks or regions of open chromatin within the specific genes, and this step retrieves the relevant annotations. Regarding getGRanges from ensemble data, this function extracts gene annotations from the ensemble object, which holds detailed gene information and converts them into genomic ranges objects from the genomic ranges package. These objects help in associating chromatin accessibility data with specific genomic features like genes or regulatory regions, and then sequence level style and genome to ensure that the genomic coordinates are compatible with the UCSC genome browser style and that the genome version is set to HG38, the human genome reference. So we write genome annotations HG38, and for annotation ppmc ATAC-seq, and we put annotation. Now let's deal with ATAC. So we exclude the first dimension as this is typically corrected with sequencing depth. So run and then TF-IDF, and then we put our data, as this technique used to normalize ATAC-seq data to adjust the differences in read coverages across cells and genomic regions. This way, highly abundant regions of open chromatin, which might have a lot of reads, are down-weighted, while less frequent but potentially biologically important regions are emphasized. And then we use function find the top features. This function to identify the most variable or informative genomic features, typically peaks. In addition, run SVD or singular value decomposition, which is a dimensionality reduction technique used for analyzing ATAC-seq data. It is similar to PCA, but is often better suited for sparse high-dimensional datasets like ATAC-seq data. And then finally, we run UMAP to perform UMAP on the ATAC-seq using SVD results, just like for rnaseq data, to reduce the dimensionality and enable visualization. So, ppmc.atac and reduction LSI dimensions from 2 to 30, and we put reduction name UMAP ATAC, reduction key ATAC UMAP. Let's run our code. Now let's plot the results from both modalities using the plot function. So we create P1, then plot, and we put our data, the RNA, group by Seurat annotations, and we make label true, no legend, G title, RNA, and we make the same for ATAC-seq data, group by reg ident, label false, no legend, and we put the title, and then P1 plus P2. Let's run our code, and here we have it. So as you can see, we have two charts, RNA and ATC. For the RNA, it shows single-cell RNA-seq data where different cells are annotated and colored. So we have different colors and we have different cells like CD16 mono, CDC, C14 mono, PDC, HSPC, and so on. This clustering is based on gene expression profiles. And here, the ATC, this represents single-cell ATAC-seq data which measures chromatin accessibility. While the clusters are visually distinct, no labels are provided. The structure should correspond to RNA clusters, but may have differences due to nature of the ATAC-seq data. So what is the purpose of the comparison? The goal is to compare how cells cluster in RNA-seq versus ATAC-seq data, assessing similarities and differences in their biological signals, identifying anchors between both datasets, as we want to connect single-cell RNA-seq or gene expression and single-cell ATAC-seq or DNA accessibility to understand how gene expression is regulated by DNA accessibility. So for gene activity, we calculate gene activity from ATAC-seq by looking at the gene body and its upstream region. This gives us a score for how active a gene might be based on DNA accessibility. And for combining the data, we combine the gene expression from RNA-seq with the gene activity scores from ATAC-seq. And for finding anchors, using canonical correlation analysis or CCA, we identify anchors. Anchors are cells where there is a correlation or similarity between RNA expression from single-cell RNA-seq and DNA accessibility from single-cell ATAC-seq. In other words, anchors represent cells with similar patterns of gene expression and DNA accessibility for the same genes. So how does CCA help? It's a statistical method used to find relationships between two datasets. In this case, DNA accessibility and RNA expression. So we quantify the gene activity, gene activity, PBMC, ATAC, features, variable features, PBMC, and then PBMC activity, and then create assay object, counts, equal gene activities. And then we normalize the gene activities. So default assay, PBMC.ATAC activity, and then normalize data, and then we scale data. And lastly, we identify the anchors. So we use find the transfer anchors function, reference PBMC RNA, query ATAC, and features, variable features, we put object equal to PBMC.RNA. For reference assay RNA, for query assay activity and reduction, we make it equal CCA. As CCA to identify cells or anchors where the RNA and the ATAC data are most similar, allowing for the integration of these two datasets. Let's run our code. After identifying the anchors, we can transfer annotations from single-cell RNA-seq onto single-cell ATAC. The output will contain a matrix with predictions and confidence scores for each ATAC-seq cell. So, in this case, we can use transfer data anchor set equal to transfer.anchors, ref data equal to PBMC RNA, $, Seurat annotations, weight reduction equal PBMC ATAC, LSI and DIMS from 2 to 30. Also for ATAC, add metadata, PBMC.ATAC and metadata cell type dot predictions. As you can see, transfer data is function used to transfer the annotations like cell type labels from single-cell RNA-seq to single-cell ATAC. Let's run our code. Now annotating single-cell ATAC-seq cells via label transfer. The goal of this step is to predict the cell types of single-cell ATAC-seq cells by using information from single-cell RNA-seq cells. We want to check if we can accurately transfer the cell type information or annotations from RNA-seq data to ATAC-seq. So we have two annotations. Predicted annotation. This is the guessed cell type for each single-cell ATAC-seq based on the pattern seen in single-cell RNA-seq data. After transferring information from RNA-seq, we can make predictions about the type of cell each single-cell ATAC-seq cell is. So, PBMC, ATC, annotation, correct, PBMC, ATC, predicted ID, and then PBMC, ATC, Seurat annotations, and we make two charts. First one, then plot, ATC, we call it predicted ID label with G title, group by predicted ID, and the title predicted annotation. and the other plot group by Seurat annotations, and gtitle ground truth annotation. And then we put P1 and P2. Let's run our code. And here we have it. As you can see, the predicted and the ground truth annotations in the chart are closely matched, including that the model accurately classifies cell types based on chromatin accessibility. So for example, here CD14 mono, and here is the same CD14 mono. Here CDC, here CDC, here CD16 mono, CD16 mono, asthma, and so on. So now we should evaluate the accuracy of our predicted annotations for single-cell ATAC-seq cells. So we write predictions, table, PBMC ATAC Seurat annotations, and then PBMC ATAC predicted ID, and then predictions, row sums predictions, to normalize for the number of cells in each cell, and predictions as.data.frame predictions, and then P1 ggplot predictions, variable 1 var2 fill equal fric, geom title, scale fill gradient, name we put fraction of cells, and then we make for low and for high, X label, cell type annotation RNA, Y label, predicted cell type label ATAC, and theme cow plot. Here we use a cow plot plus theme, and here correct length, which PBMC ATAC Seurat annotations PBMC ATAC predicted ID, and for the incorrect length, which is PBMC ATAC Seurat annotations doesn't equal to, or it's not PBMC ATAC $ predicted ID. And for data, we use fetch data, PBMC ATAC, and prediction score maximum, and annotation correct. And for the plot 2, ggplot2 data, prediction dot score max, fill annotation correct, color annotation correct, geom density, alpha 0 or 0.5, alpha equal to 0.5, theme cow plot, and we scale fill discrete, name equal annotation correct, and we put the labels, and after that we collect the plots together, P1 plus P2. Let's run our code. We can make our chart bigger. So as you can see, predicted cell type ATAC, cell type RNA, and the prediction often it's 90% or over 90%, as it's very dark brown. And this lighter brown, it's about 60 to 65%. So as you can see, this heat map shows the fraction of cells assigned to each predicted cell type ATAC versus RNA. There is a strong suggestion that the most predicted labels match the ground truth. And for this plot, or the right plot, the x-axis is the prediction score, while y-axis is the density. The density plot displays the distribution of predicted scores for correctly or the blue and incorrectly the red. Here's a false and here's a true. The correct predictions tend to have higher score, closely to 1, while incorrect predictions are closely frequent at lower scores. So here we are closer to 1 for the true, while the false is about 0.5, and so on. And now, co-embedding single-cell RNA-seq and single-cell ATAC-seq datasets. In order to perform co-embedding, we first impute RNA expression into single-cell ATAC-seq cells based on the previously computed anchors, and then merge the datasets. So, gnews equal to variable features PBMC RNA, and reference data or ref data, get assay data PBMC RNA, assay equal RNA, slot data, and we put gnews. The ref data contains single-cell RNA-seq expression matrix for single-cell RNA-seq cells imputation. The output will contain an imputed single-cell matrix for each ATAC cells. So, imputation equal to transfer data anchor set transfer anchors, ref data, or equal to ref data, weight reduction, PBMC, ATAC, DIMS 2 to 30, ATAC, RNA equal to imputation, co-embedded, merge, X equal PBMC.RNA and Y PBMC ATAC. Finally, we run PCA and new map on the combined object. So we scale the data and then run PCA, run new map, and then dim plot. We put the co-embed that we created, group by C, org ident, Seurat annotations. Let's run our code. And here we have it. We got the two plots, org ident, and Seurat annotations. UMAP1 versus UMAP2. Here is the same. For the red color for ATAC, and the blue is the RNA. And here for Seurat annotations, composed of the cells, different cells, each cell with different color. X-axis UMAP1, Y-axis is UMAP2. And here we've reached the end of our tutorial. I will leave the links in the description of the videos that help us to do this analysis. If you enjoyed this tutorial, please don't forget to like, share and subscribe for more content. Thanks for watching, see you next time.